Dear student, welcome you all in the virtual process of learning. Today we will learn about qualitative test for amino acids and protein. Qualitative analysis designed to identify the component of a substance or mixture. Qualitative analysis also tell us whether an atom, ion, functional group or a compound is present or absent in a sample, but it does not provide information about its quantity. Quantification of a sample is in contrast is called quantitative analysis. So in today's lecture we will learn qualitative analysis of amino acid and protein. There are number of tests to detect the presence of amino acids and protein. This is depend on the natural of side chain. In today's lecture, we will learn about hydrogen test that is used for the alpha L amino acids, biuret test that is used for identification of presence of peptide bond, xanthopratic test that is used for aromatic amino acids, lead sulfide test that is used for the detection of amino acids containing sulfur atom. Millens test used for the amino acid containing hydroxyphenyl group in their structure. Sakaguchi test and the Hopkins-Colt test that is useful for the identification of amino acid tryptophan. Xanthoprotic test. Some amino acids containing aromatic group that are derivative of benzene. These aromatic groups can undergo reaction. One such reaction is the nitration. Nitration of a benzene ring with nitric acid, the amino acid that have activated benzene group can readily undergo nitration. This is the basic principle behind the xanthoprotic test. Objective is to differentiate between the aromatic amino acids which give positive results, basically the yellow color, and other amino acids. So, xanthoprotic test is used for identification between aromatic amino acids and other amino acids. What is the principle? Concentrated nitric acid react with aromatic nucleus present in the amino acid side chain gives the nitration reaction giving the solution yellow color. Amino acid tyrosine and tryptophan containing activated benzene ring. Basically, they having the heterocyclic nucleus which are readily nitrated to give yellow color compound. While the aromatic ring of phenylalanine does not react readily with nitric acid despite it contains a benzene ring, but it is not activated, therefore it will not react and do not give yellow color on in xanthoprotic test. These are the chemical reaction of tyrosine, tryptophan and phenylalanine with nitric acid. Phenylalanine still react with nitric acid, just not as readily as tyrosine or tryptophan, because tyrosine and tryptophan have electron donor hydroxyl and the nitrogenous nitrogen heteroatom, which make the ring a lot more reactive for electrophilic nitration. To get phenylalanine to react, you may have to boil it with the nitric acid that gives yellow color after heating. Lead sulfide test. Sulfur containing amino acid, for example cysteine and cysteine, upon boiling with the sodium hydroxide, that means hot alkali, yield sodium sulfite. This reaction is due to partial conversion of organic sulfur to inorganic sulfide which can be detected by precipitating to the lead sulfide using lead acetate solution. The objective of this test is used specific for sulfhydryl group containing amino acid. Principle is sulfur in cysteine or amino acid is converted to sodium sulfide by boiling with 10% NaH solution. The sodium sulfide can be detected by precipitation of lead sulfide from an alkaline solution when adding to lead acetate. Minion test. Phenolic amino acid such as tyrosine and its derivative respond to this test. Compound with a hydroxybenzene radical react with Minion's reagent to form a red color complex. Minion's reagent is a solution of mercury sulfate in the sulfuric acid. So objective of this study is this is the particular test used for the identification of tyrosine because it is the only amino acids containing a phenol group. Principle. The phenol group of tyrosine is first nitrated by nitric acid in the test solution. Then the nitrated tyrosine complex mercury ions in the solution to form a brick red appearance of red color is the positive test. The next test is the Sakaguchi test. This test is used for identification of amino acid containing guanidine group. In other words, we can say that this test is used for identification of arginine amino acid. Principle. In the alkaline solution, arginine react with alpha naphthol and sodium hypobromite or chloride as an oxidizing agent to form a red complex as a positive result. This is the result. This is the test performed for the glycine and test performed for the arginine. Arginine amino acid gives red complexes as a positive. The next test used for identification of amino acid is Hobskin-Colt reaction. This test is specific for tryptophan, the only amino acid containing indole group. Principle. The amino acid react with glycolic acid in the presence of strong acid. The indole ring forms a violet cyclic product. The protein solution is hydrolyzed in the concentrated sulfuric acid in the solution interface. Once the tryptophan is free, it reacts with glycolic acid to form violet complex product. So this test is particularly used for indole containing amino acid. Summary of today's lecture. Qualitative analysis designed to identify the component of a substance or a mixture. Qualitative analysis also tell us whether an atom, ion, functional group or compound is present or absent in a sample. There are various tests used for the identification of amino acid and protein in a sample. These are, the first one is Nin-hydrin test. That is a universal test for all amino acids and protein. A free the amino acid containing free amino group gives this test positive. Nin-hydrin test gives a violet color with the nin-hydrin reagent. Biority test. This test is used for the identification of peptide. A violet blue color complex observed after the amino acid or protein react with biuret reagent. The next test is the xanthoprotic test. This test is used for the identification of amino acid having aromatic in their side chain. Xanthoprotic In the xanthoprotic test, the amino acid on reaction with nitric acid gives yellow color. This yellow color is due to the uh, production of nitration of activated benzene. Lead sulfide test. This test is used for the detection of amino acid containing sulfur hydryl group. In this test, the cysteine is first reacted with sodium hydroxide which make sodium sulfide, which on reaction with lead acetic acid gives the precipitate of lead sulfide. Maillon test. This test is used for the identification of amino acid containing hydroxyphenyl group. In this, nitration of tyrosine complex in the mercury ion in the solution to form a brick red. This test, Maillon test, is used for basically for the tyrosine amino acid. Sakaguchi test. In this test is particularly used for the identification of amino acid having guanidine ring. So, asparagine is the amino acid which is having guanidine ring. Hopkins Call test. This test is used for indole containing amino acid. Tryptophan is the example. The tryptophan reacting with the glycolic acid in the presence of strong acid, the indole ring forms a violet cyclic product. So these are the various color reactions used for the qualitative analysis of amino acids and amino acids present in the proteins. In this slide, we will learn about the pattern of question asked in your examination. So there are three patterns. First one is the long answer, second one is the fill in the blanks, and third one is the MCQ. So in the long answer, the example is describe the various tests used for the identification for amino acid. So you have to explain all the amino acids reaction used for identification. For example, in hydrogen test, Genthoprotic test, Maillon test, Hopkins Call test, and lead sulfide test, and so on. In next question is you have to write down the fill in the blanks. Name the test used for the identification of amino acid containing the sulfur hydryl group. We have in the in this lecture we already learned that a lead sulfide test is used for identification of amino acid having the sulfur hydryl group. So next question, Genthoprotic test used for the identification of amino acids containing aromatic amino acids. Hopkins Call test is used for the identification of, you have given the, there is the four option, you have to choose the right option. The right option is the tryptophan. Protein on reaction with bioreagent gives the fill in the blanks color. So you have to choose the right color. The answer is the violet color. So, dear student, if you want more information, you can refer to these books, YouTube, and this website. Thank you, students, for becoming a fairy participant. If you have any issue or query, you can text me or you can mail me. We will meet again. Till then, namaste.